Imagine trying to capture the sound of a heartbeat in a noisy subway station. How do you know if what you're recording is real or just background chaos?
That's the heart of the challenge in physiology, measuring real biological signals while cutting through all the interference. Hi everyone, I'm here with you to break down the basics of measurement and analysis in physiology.
Today, we'll unravel how scientists decide what's a signal and what's noise, why measurement models matter, and how to ensure the info you get is actually useful.
We'll explore the role of models in measurement, handling noise, the anatomy of measurement systems, and what makes a good sensor. Ready to dive in?
Funny thing, before you even start measuring, you need a mental picture of what you expect to find. Like, you can't measure blood pressure meaningfully unless you know it should rise and fall with every heartbeat.
Exactly, and that model shapes what counts as the actual signal versus what's just noise. If we're recording heart sounds, traffic outside is noise. But if we're analyzing traffic, that same sound becomes our signal.
So it really depends on your goal. Your model helps you decide what stays and what goes, which is kind of wild when you think about it.
Let's make it concrete. Imagine recording music at a concert. The song is your signal, but the crowd chatter? That's noise, and you want it gone.
But what if you're doing a study on audience reactions? Suddenly, that chatter is the star of the show, and the music is just background. Perspective totally changes what you value.
Which is why the first step in measurement is getting clear on your model and what you actually want to analyze.
All right, let's talk about actually recording physiological signals. It sounds simple. Hook up a sensor and you're done. But there's so much more to it.
Right, because the measured signal always contains some unwanted interference. Good setup, like using the right kind of microphone or electrode, can make a huge difference in how much noise sneaks in.
And if you can't avoid noise completely, you have to be ready to clean it up later using signal processing.
Once you've got your signal, your interpretation really depends on your underlying model. For instance, if you see pressure in a blood vessel fluctuating, that's normal.
But if the same happens in a water pipe, it signals a problem. So, understanding the system you're working with is everything otherwise. You might misinterpret what you're seeing.
Measurement is more than just gathering numbers. It's about extracting meaningful patterns. Sometimes, models even help compress data so doctors can spot issues faster.
Yeah, like using an ECG model to classify heart conditions. Instead of combing through raw voltage data, the system narrows things down to key parameters for easier diagnosis.
That's where research and clinical practice split. Researchers develop models, while doctors use them to analyze patient data.
Let's zoom in. A signal is just any quantity that changes over time or space. For the heart, it's voltage over time. For a photo, it's light intensity across a surface.
You can even have three-dimensional signals, like tissue density in 3D space, or a movie, where the scene changes both in space and over time. The number of independent variables gives the signal its dimension. Let's untangle what happens from start to finish. It all begins with the physiological process, say, muscle contraction producing a force.
That force gets picked up by a transducer, maybe a load cell that converts force into an electrical voltage. Then, it's amplified, filtered, and finally displayed or digitized for analysis.
Each block in that chain modifies the signal in some way, and their combined effect is called cascading.
Transduction is a fancy word for turning one type of energy into another. Think of a thermometer. Heat turns into the movement of mercury.
Most modern systems convert everything into electricity, voltage, or current because it's so much easier to process with instruments or computers.
But here's the catch. No transducer is perfect. They can add distortion, noise, or just respond too slowly.
Noise can sneak in from other physiological processes, electronics, or even electromagnetic fields nearby. Sometimes the noise is random, like thermal jitter, or rhythmic, like the 50 Hz hum from power lines.
And it's almost always mixed in with your signal, which makes separating them tricky. Sometimes their properties overlap so much that removing noise means losing part of your real signal too.
Whenever possible, we try to prevent noise at the source. Good electrode placement, shielding, and careful setup help a lot.
But if noise still gets through, we use filtering either analog hardware or digital software. But remember, every filter involves a trade-off. Remove too much noise, and you might erase valuable data.
That's why knowing your signal's unique features is key. The clearer the distinction, the better you can preserve what matters.
Let's get into sensor calibration. Static calibration is about how the sensor responds to steady inputs, a thermometer in a water bath, for instance.
Dynamic calibration checks how fast and accurately a sensor tracks changes, like how quickly a thermometer reacts to a temperature jump.
Ideally, sensors respond linearly, double the input, double the output. But in reality, every sensor has limits. If you push it too far, it can distort or even saturate the signal.
Sometimes, you can use math tricks like linearization to correct for nonlinearity, but it's always better to work within the sensor's sweet spot whenever possible.
To test a sensor's speed, we use step or sinusoidal signals. A step input, like dunking a thermometer in hot water, lets us see how quickly it catches up.
And by analyzing its response, we can calculate things like time constants or natural frequencies, which tell us how well the sensor will track real-life, fast-changing signals.
Let's wrap it up with a classic, the ECG. Multiple electrodes measure voltage differences across the body, but noise, like power line interference, is always lurking.
Differential measurement and clever electrode placement help cancel out common noise. Then, using the Einthoven triangle model, we can reconstruct the electrical activity of the heart in space and time.
And that's the magic of model-based physiological measurement, combining hardware, math, and physiology to uncover what's really happening inside the body.
Let's highlight 3 key takeaways. First, physiological measurement always relies on a clear model of your system. Second, noise can never be ignored. Its reduction demands strategy and sometimes compromise. Third, the characteristics of your sensor shape the quality of your data.
If you're just getting started, here's your action step. Next time you set up a measurement, sketch out your system model. Map out what's signal, what's likely noise, and plan how you'll handle both before you ever hit record.
That way, you'll be ready to get the cleanest, most meaningful signals and make sense of the body's hidden rhythms. Thanks for listening, and stay curious.